In this unit, we're talking about the chromosomal basis of inheritance. And so, first, Mendel came up with two laws. One of them is the Law of Independent Assortment that says that every possible combination of alleles is equally likely for each gamete. So in this picture, I've got circled all of the possible combinations of alleles that could come from this potential metaphase division. So we've got red with red alleles up here. We've got blue with blue alleles over here. We've got red and blue over here, and blue and red over there, which ends up being the same thing. The second of Mendel's laws is the law of segregation. This says that paired genes separate and randomly recombine into the gametes so that offspring have an equal likelihood of inheriting either one. So that brings us to concept of probability. If independent assortment is occurring, the laws of probability predict the genotypes of offspring from the crosses. So the parental generation, which we show with big P, is the parents of a cross between two individuals. The first filial generation, which we represent with F1, is the offspring of P. The second filial generation, which we show with F2, is the offspring from the cross between two F1 individuals. So, for instance, in this picture, our P generation is a cross between a pea plant with white flowers and a pea plant with purple flowers. We happen to know the genotypes of these two pea plants here. The white-flowered pea plant is homozygous recessive, and the purple-flowered pea plant is homozygous dominant. So when we cross these two plants, what we end up with is the F1 generation, down here. 100% of the individuals are purple, and all of them have a heterozygous genotype. And then, when we cross two F1 individuals, you get the F2 generation, and your individuals can either be little p, big P, or big P, little p. And so we use something called a Punnett Square to estimate the probability or the likelihood of getting one genotype in the offspring or another, which then tells us the probability of getting one phenotype or another. So we use the gametes that each individual could possibly produce to see the predicted offspring's genotypes and phenotype ratios. So over here, I've drawn a Punnett square that shows you the results of a cross between two individuals that are both heterozygotes. So the mother plant, so to speak, could produce a big Y egg or a little y egg because her genotype is big Y, little y. Now the father plant, so to speak, could also produce a big Y sperm or a little y sperm because his genotype is big Y, little y. And so, using this, we combine down the columns and across the rows to see the possible genotypes of offspring. So, when we combine the big Y from the mother with the big Y from the father, we get a big Y offspring. When we combine the big Y from the father with the little y from the mother, we get a big Y, little y. And so on, so that you end up with big Y, little y, and finally one that is both little y, little y. So check your supplemental readings to learn more about Punnett squares and probability and predicting even more complicated crosses than those with just two characteristics.